homologous to the basic histone proteins of eukaryotic cells. Similar DNA-binding histone-like proteins have been found in several hyperthermophilic eukaryota. Ferroplasm in the chemolithotrophic relative to thermoplasm. Like thermoplasm, it lacks a cell wall and has an acidophilic lifestyle. It is not a thermophile, growing optimally at 35 degrees Celsius. Ferroplasm oxidizes Fe2 to Fe3 to obtain energy. This reaction generates acid. And see legend to figure 13.15 and uses CO2 as its carbon source autotrophy. Ferroplasmic grows in mine tailings containing pyrite FeS. Its energy source, the extreme acidophily of ferroplasm, allows it to derive down the pH of its habitat to extremely low values. It is believed that after moderate acidity is generated from Fe+, 2+, oxidation by organisms such as Thiobacillus ferrooxidans and Leptospirillum ferrooxidans, ferroplasm becomes active as subsequently generates the very low pH values typical of acid mine drainage. Phycophilus. Phylogenetic relative of thermoplasm and thermoplasma and ferroplasma is Phycophilus. Although thermoplasm and ferroplasm are extreme acidophiles, Phycophilus is even more so, growing optimally at pH 0.7 and capable of growth to as low as pH negative CO 0.06. Pyrophilus differs from the thermoplasm and ferroplasm in other ways as well, including having a cell wall, and has a much lower DNA GC base ratio. Two species of Pyrophilus have been isolated from the acidic Japanese sulfurous Sulfiteris, and the thermoplasm both grow heterotrophically on complex media. The physiological in Pyrophilus is clearly of interest as a model for acid tolerance. Studies of its cytoplasmic membrane suggest an unusual arrangement of lipids that form an extremely acidic, impermeable membrane at optimal pH values. By contrast, its only moderately acidic pH such as pH 4, the membranes of cells of Pyrophilus quickly become leaky and literally disintegrate, clearly indicating that this organism has evolved to survive only in high acidic habitats. Hyperthermophilic Euarchaeota and thermococcales and methanopyrates. Thermococcus, Pyrococcus, and methanopyrates. A few euarchaeotes thrive in a thermal environment, and some are extremely thermophilic. Those with growth temperature optima over 80 degrees Celsius are called hyperthermophiles. We consider there are three hyperthermophilic eukaryotics that branch on the archaea tree, very near to the roots. Two of these organisms, Thermococcus and Pyrococcus, show phenotypic properties very similar to those of hypothermophilic crinoarchaeotes to be discussed. And form of distinct order, the thermococcales. The other methanopyrus is a methanogen and closely resembles the methanogens in its basic physiology, but it's unusual in its extreme requirements for heat and phylogenetic position, distinct from that of other methanogens. Thermococcus and Pyrococcus. Thermococcus is a spherical hypothermophilic eukaryota indigenous to anoxic thermal waters in various locations throughout the world. The spherical cells contain a tuft of polar flagella and are thus highly motile. Thermococcus is an obliquely anaerobic chemoorganotroph that grows on proteins and other complex organic mixtures, including some sugars with SO to the power 0 as electron acceptor of temperatures from 70 to 95 degrees Celsius. Pyrococcus is morphologically similar to Thermococcus. Pyrococcus, of Latin derivation, literally means fireball, differs from the Thermococcus primarily by its higher temperature requirements. Pyrococcus grows between 70 and 106 degrees Celsius, with an optimum of 100 degrees Celsius. Thermococcus and Pyrococcus are also metabolically quite similar. Protein, starch, or maltose are oxidized as electron donors, and S to the power of 0 is terminal acceptor and is reduced to H2S. More properties of Thermococcus and Pyrococcus are described in Table 13. Methanopyrus. Methanopyrus is a rod-shaped hyperthermophilic methanogen that has been isolated from sediments near submarine hydrothermal vents and from the walls of black smoker hydrothermal vent chimneys. For a discussion of thermal vents, methanopyrus occupies a unique phylogenetic position on the tree of Archaea. It is one of the most ancient least derived of all known hyperthermophilic archaean and shares phenotypic properties with both the hyperthermophile's growth temperature maximum, 110 degrees Celsius, and the methanogens it carries out, the reaction of 4H2 plus CO2 to CH4 plus 2H2O. Methanopyrus produces methane from H2 plus CO2 only at grows rapidly generation time less than 1 hour, at its temperature optimum of 100 degrees Celsius. Unlike mesophilic methanogens, however, the cells of methanopyrus have large amounts of glycolytic derivatives, cyclic 2,3-diphosphoglycerate, dissolved in the cytoplasm. This compound, present at more than 1 m concentration in methanopyrus, is thought to function as a thermal stabilizing agent to prevent denaturization of enzymes and DNA inside the cell. In addition, methanopyrus contains a type of membrane lipid found in no other known organism. This ether lipid is an unsaturated form of the otherwise saturated diphenyl tetraethers found in other hyperthermophiles. The discovery of the methanopyrus may explain the origin of hydrocarbon-like materials in hot oceanic sediments previously thought to be too hot to support biogenic methanogenesis. In addition, at the depth of which methanopyrus was found, approximately 2,000 meters, water remains liquid at approximately 2,000 meters. Water remains liquid at temperatures up to 350 degrees Celsius, suggesting that other hyperthermophilic methanogens may exist capable of growth at 110 degrees Celsius or at perhaps even higher temperatures. Archaeoglobus and Ferroglobus. We will see later that a number of hyperthermophilic Cree and Archaeota carry out the anaerobic respiration in which S to the power of 0 is used as an electron acceptor being reduced. To H2S. Curiously, none of these organisms are sulfate reducers that is capable of reducing SO4 to H2S. However, one hyperthermophilic eukaryotic Archaeoglobus is true sulfate reducer and forms of phylogenetically distinct lineages within the Eukaryota. Archaeoglobus and its genome. Archaeoglobus and its genome. Archaeoglobus was isolated from the hot marine sediments near hydrothermal vents and couples the oxidation of H2 lactate pyruvate glucose or complete organic compounds to the reduction of sulfate to sulfide. Cells of Archaeoglobus are irregular cocci, and cultures grow optimally at 83 degrees Celsius. However, one of the most remarkable features of Archaeoglobus is its relationship to methanogens. We will learn in Chapter 17 about the unique biochemistry associated with the methanogenesis. Briefly, this involves the use of novel coenzyme-like factors and many others. Thus far, these novel biomolecules have only been found in cells of methanogens. Surprisingly, however, Archaeoglobus contains traces of these coenzymes, and cultures of this organism actually produce small amounts of methane during growth. Thus, Archaeoglobus, which also shows a rather close phylogenetic relationship to methanogens, may represent an evolutionary transitional type of organism among the archaea, one that bridged the energy-generating processes of sulfur reduction and methanogenesis. More information on the methanogenesis by Archaeoglobus has energy from the complete genome sequence of this organism. The sequence has revealed that although the Archaeoglobus can make methane, it lacks genes for the key enzyme of methanogenesis found in methanogens, methyl-CoM reductase. Thus, the small amounts of methane that are made by Archaeoglobus do not come from the activity of the enzyme, and their exact origin is unclear. The genome of Archaeoglobus contains about 2,400 genes, and interestingly shares a number of genes with species of methanogens. However, this Archaeoglobus genome sequence also reveals a number of unique genes. This suggests that genetic diversity in the archaea is quite intensive, and that analysts of the genome of archaea will likely mimic the findings of the genome project of various bacteria. Each new organism sequenced has about 1/4 of its genes, a unique gene never before seen in other organisms. Ferroglobus. Ferroglobus is related to Archaeoglobus, but is not a sulfate-reducing bacteria. Instead, Ferroglobus is an iron-oxidating chemolithotrophic, autotrophic conserving energy from the oxidation of Fe2+, coupled to the reduction of NO3, plus NO. Ferroglobus can also use H2 or H2S as electron donors in its energy metabolism. Ferroglobus was isolated from the shallow marine hydrothermal vent and grows optimally at 85 degrees Celsius. Ferroglobus is interesting for several reasons, but especially so because of its ability to produce Fe to the power of 3+ from Fe to the power of 2+ under anoxic conditions. The mechanism for the formation of Fe to the power of 3+ in ancient rocks previously assumed to have been from the oxidation of Fe to 2+ by O2 produced by cyanobacterium is now being questioned with the discovery that anoxic routes to the production of Fe to the power of 3+ exist from the activities of organisms like Ferroglobus and carbon and oxygen.